Namaste, Hamisha खुश रहो बिटिया. My name is Manisha Singh. Today I am teaching you storage of food at chilling temperatures in subject Principles of Food Preservation Technology under Food and Biotechnology discipline. First of all, what is what is the need of the preservation by low temperature? The use of low temperature is the easiest, most convenient, and least time consuming method of preserving foods. Refrigeration and freezing do not sterilize foods or destroy the microorganisms that cause spoilage. It just simply slows the growth of microorganisms and chemical and enzymatic changes that affect quality and cause spoilage. Low temperature preservation is less effective as compared to the thermal techniques of food preservation because microorganisms are most likely to be able to survive cold temperatures than the hot temperatures. One of the problems related to the use of freezing as a method of food preservation is that microorganisms are only deactivated and not killed, which may again become active while thawing. And similarly, enzyme action is slowed but not stopped during freezing, and these enzymes are responsible for color and flavor changes and loss of nutrients during refrigerated storage. Chilling techniques. Chilled foods are those foods stored at temperatures near but above their freezing point, typically 0 to 5 degrees Celsius. This commodity area has shown a massive increase in recent years as traditional chilled products such as fresh meat, fish, and dairy products have been joined a huge variety of new products including complete meals. Then refrigerated storage of food is generally practiced both at home or at industrial level. In chilling, the temperature of a food is reduced generally to between 1 degree Celsius to 7 degree Celsius. And thus subsequent storage at refrigerated temperature extends the shelf life of both the fresh and processed foods. It just increases shelf life and has limited applications in the preserving foods. It is not the sole method for preserving food, therefore, it is used as an adjunct process to extend the storage life of mildly processed and low acid foods. Chilling and refrigerated storage retards the growth of bacteria and particularly the thermophiles and the mesophiles. Psychrophilic spoilage bacteria however, can spoil food during low temperature storage, but psychrophilic pathogens such as Listeria monocytogenes, Yersinia enterocolitica, etc., found in refrigerated food need attention. Then temperature of 5 to 6 degrees Celsius or less retard the growth of most food poisoning microorganisms except Clostridium botulinum type E. There are three main factors have contributed to this development are the food manufacturer's objective of increasing added value to their products, consumer demand for fresh foods while at the same time requiring the convenience of only occasional shopping and ease of preparation. The availability of an efficient cold chain, the organization and infrastructure which allows low temperatures to be maintained throughout the food chain from manufacture or harvest to the consumption. Chill storage can change both the nature of spoilage and the rate at which it occurs. There may be a qualitative changes in spoilage and characteristics as low temperatures exert a selective effect preventing the growth of mesophiles and leading to a microflora dominated as psychrotrophs. This can be seen in the case of raw milk which in the days of milk churns and roadside collection had a spoilage microflora comprised largely of mesophilic lactococci which would sour the milk. Nowadays, in the UK, milk is chilled almost immediately. It leaves the cow so that psychotrophic gram-negative rods predominate and produce an entirely different type of spoilage. Factors can also cause physiological changes in microorganisms that modify or exacerbate spoilage characteristics. The such examples are the increased production of phenazine and carotenoid pigments in some organisms at low temperatures and the stimulation of extracellular polysaccharide production in leuconostoc species and other lactic acid bacteria. In most cases, such changes probably represent a disturbance of metabolism due to differing thermal coefficients and activation energies of the numerous chemical reactions that comprise microbial metabolism. Though psychotrophs can grow in chilled foods, that they do so only relatively slowly so that the onset of spoilage is delayed. In this respect, temperature changes within the chill temperature range can have pronounced effect. For example, the generation time for some pseudomonad isolated from fish was 6.7 hours to 5 degrees Celsius compared to 26.6 hours at 0 degrees Celsius, where this organism is an important contributor to the spoilage. Small changes of temperature will have major implications for their shelf life. The keeping time of haddock and cod fillets has been found to double if the storage temperature is decreased from 2.8 degrees Celsius to 0.3 degrees Celsius. The mathematical modeling techniques of the short can be useful in predicting the effect of temperature fluctuations on shelf life, but as a general rule, storage temperature should be as low and as tightly controlled as possible. The ability of organisms to grow at low temperatures appears to be particularly associated with the composition and architecture of the plasma membrane. As the temperature is lowered, the plasma membrane undergoes a phase transition from a liquid crystalline state to a rigid gel in which solute transport is severely limited. The temperatures of this transition is lower in psychotrophs and psychrophiles largely as a result of higher levels of unsaturated and short-chain fatty acids in their membrane lipids. If some organisms are allowed to adapt to growth at lower temperatures, they increase the proportion of these components in their membranes. There seems to be no taxonomic restrictions on psychotrophic organisms which can be found in the yeasts, molds, gram-negative and gram-positive bacteria. One feature they share is that in addition to their ability to grow at low temperatures, they are inactivated at moderate temperatures. A number of reasons for this marked heat sensitivity have been put forward, including the possibility of excessive membrane fluidity at high temperatures and low thermal stability of key enzymes and other functional proteins appears to be an important factor, although thermostable extracellular lipases and proteases produced by psychotrophic pseudomonads can be a problem in the dairy industry. Though the mesophiles cannot grow at chill temperatures, they are not necessarily killed. Chilling will produce a phenomenon known as cold shock, which causes death and injury in a proportion of the population, but its effects are not predictable in the same way as heat processing. The extent of cold shock depends on number of factors such as organism, that is gram negatives, appears more susceptible than gram positives, its phase of growth, and the temperature differential and the rate of cooling, and the growth medium cells grown in complex media are more resistant. The principal mechanism of cold shock appears to be damage to the membranes caused by the phase changes in the membrane lipids, which can create hydropores and through which cytoplasmic contents can leak out. An increase in a single strand breaks in DNA has also been noted, as well as the synthesis of specific cold shock proteins. Since chilling is not a bactericidal process, the use of good microbiological quality raw materials and hygienic handling are key requirements for the production of safe chill foods. Mesophiles that survive cooling and albeit in an injured state can persist in the food for extended periods and may recover and resume growth should conditions later become favorable. Thus, chilling will prevent an increase in the risk for mesophilic pathogens that will continue to grow at some chill temperatures, and they key roles of chilling in the modern food industry has focused particular attention on these. Risks posed by these organisms may increase with duration of storage, but this process is likely to be slow and dependent on the precise storage temperature and composition of the food. Some foods are not amendable to chill storage as they suffer from cold injury, where the low temperature results in tissue breakdown, which leads to visual defects and accelerated microbiological deterioration. Then tropical fruits are particularly susceptible to this form of damage. Hope you all are satisfied with this session. This session is powered by Digital Version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, please mention it in a comment box. I will resolve them whenever we have a live session. Thank you so much for watching this video.